Thank you everyone for coming to the Rare Disease Clinical Trials Network Journal Club. It's great to be back. And we have two presentations, so there'll be about a 20-minute presentation and then a 5 to 10-minute Q&A. And just to re-emphasize, if you put your questions into the Q&A tab, and then we will read out those questions and put it out for both the panelists as well as the general for discussion. So to start off with, we have Kevin Gaughan. He's a pediatric specialist registrar in CHI, and he is going to be giving us a talk on efficacy and safety of a synthetic biotic for treatment of phenylketonuria, a phase 2 clinical trial. And Kevin has a special interest in metabolic and rare diseases. So it's a pleasure, and we thank him very much for this presentation. Over to you, Kevin.
Thank you very much, Dr. James, and everyone for this opportunity to present at today's Journal Club. I'll try and keep it to the 20 minutes. But yeah, as said, so I like to present on this paper published back in October of last year, looking at the use of synthetic biotics for treating phenylketonuria or PKU. There we go. So in terms of how the presentation will run, so I'll just do a quick run through in terms of a background of PKU, particularly for those outside the field of metabolic disorders. We'll talk about the study and how it was designed, as well as the results, obviously, and then have a brief kind of review of the paper and discussion. So PKU is an inherited metabolic disorder caused by a defect of an enzyme, in this case phenylalanine hydroxylase, which is responsible for converting phenylalanine to tyrosine. It's an autosomal recessive condition, and it's actually basically one of the most, or if not the most prevalent inborn error of metabolism. The prevalence varies globally, kind of anywhere to around 1 in every 20,000 live births globally, but it's quite common in Ireland, and the prevalence in Ireland is about 1 in every 4,500 live births. If PKU is left untreated, it can lead to a moderate to severe phenotype with physiological and neurological disabilities, which are irreversible long term. This is the enzyme pathway that's affected in PKU. So phenylalanine being converted to tyrosine, which, when is unable to do so, phenylalanine accumulates and causes downstream toxic effects. There's a cofactor involved, which I've left here because I'll be mentioning it later, biopterin, which is involved in this pathway as well. We'll come back to that later. I thought it'd be worth talking briefly about the history of PKU, because it kind of leads us up to where we are currently in terms of managing patients with PKU. The disorder was first kind of described in the 1930s by a Norwegian physician named Dr. Volling. Basically, there were two parents who approached him, had 3 and 1, who both had progressive intellectual disability. And when he analyzed their urine, he found that there was phenylpyruvic acid and phenylketones in the urine, and that's where the name PKU, or phenylketonuria, came from. With the discovery of that, there was a rush to see were other children with intellectual disability also PKU, and if so, what could we do about it? In the 1950s, that led to the development, particularly by a physician in Birmingham Hospital named Prof. Bickel, for a diet that was low in phenylalanine, because we knew that if it was an issue of metabolizing phenylalanine, if we could reduce how much they were exposed to, would we reduce the condition? And that seemed to be the case. But the issue was you had to get this diet commenced early, because if you started it, say, in an adult, they're already affected. You couldn't reverse the phenotype. And that led to the introduction of the Guthrie test in the 1960s, and that spawned the newborn screening programs, which are now performed globally, and for a number of conditions, including PKU. In the 1980s, we discovered the gene for PKU. And then subsequently, we identified that patients with PKU were responsive when you actually gave them this biopterin cofactor, and that spawned the development of the drug Kuvan, which I'll talk about briefly. And then more recently, in 2018, pegvalase, which is an enzyme therapy, was approved by the FDA. So at present, how we treat PKU patients is very much centered around restricting phenylalanine phenylalanine, which I'll refer to as briefly Phe, in the diet, and making sure that they don't get deficient in other amino acids by giving synthetic protein as needed. The two additional therapies that we now have in the last 2 decades are Kuvan and pegvalase. Kuvan is a synthetic form of that cofactor. So the idea being is that by giving the synthetic cofactor, you stabilize the enzyme and increase the activity so that they're less, they're more able to tolerate phenylalanine than the diet. The issue with this is that only a portion, less than half of PKU patients actually are responsive to Kuvan or actually will benefit from it. And unfortunately, those who tend to have more severe forms of PKU are less likely to be responders. So it's a medication that's great, but it's great for patients that unfortunately are a small cohort of the overall patients that we see. Pegvalase is a subcutaneous injection therapy that is approved for adults, so it's not approved for pediatric patients. And while it does have benefits, there are adverse effects. So there's a risk of allergic reactions and anaphylactic risks that is known. So again, it's an issue in terms of for our pediatric patients, it's not one that's available. We know from kind of survey studies and review of adult patients that as patients age and they get into the adult groups, that we see a reduction in compliance and adherence to the restricted diet. And we see this in the sense that pediatric patients are multiple times more likely to have levels in control in terms of that we like to see kind of say 180 to 360 millimoles per liter, like a set range that we know is safe, where adult patients are less likely to. We know that this seems to be associated with issues in terms of an adulthood of executive function, depression, attention deficit disorders that we see more commonly in the PKU patients than with the adult patient population would, even despite good control in the younger age. And we know that patients, while they're happy, a good chunk are happy with the current treatment. About a third say that it's hard, it's difficult, there's still challenges with it, and they're looking for new therapies or hoping for new therapies to come down the line. And so I suppose in that environment, that kind of sparked the idea for new treatments, and one of which is this idea of a synthetic biotic. So a synthetic biotic is a bacterial strain that's basically been engineered, and I suppose specifically genetically engineered, to perform a function, say an enzymatic function in this case. The authors of this paper in previous work that they published before this trial developed a E. coli strain that expressed basically phenylalanine enzyme. So an enzyme that could basically kind of take over or help in terms of breaking down phenylalanine in the diet or in the gut. I mention here just because it's important when we look at the results of the paper is that when the phenylalanine is broken down, it produces metabolites, and these are TCA and hippuric acid. These come up later in terms of when they're looking to see, does this actually work for the patients and are they seeing a good result? So we'll come back to those later. Before this phase 2 study, there were 2 phase 1 studies. There's 2 different strains that are looked at in this paper. There's the 1618 and 1934. And both had phase 1 studies that show that they're safe when used for adult volunteers, both healthy volunteers and participants that had PKU. The reason for the 2 strains is that the first was developed and it seemed to be effective, but then a subsequent was developed that actually seemed to have more effective enzyme activity. So they wanted to try both to see if one was superior and to go further with. This is just a kind of graphical representation of what the bacteria is doing. It has the transporter to bring in the phenylalanine, and then that's metabolized to TCA. I suppose, globally speaking, the idea behind this is that to try and compensate for the body's inability to break down phenylalanine, can this be done in the gut with this synthetic biotic? And that's the hypothesis going into this trial. So this is the Symphony study. The goal, again, was to test the safety and efficacy of these 2 synthetic biotics in adults with PKU. The primary outcome was looking at a change of labeled phenylalanine on day 14 of treatment compared to pre-treatment. So the idea, and I'll kind of go into it with the figures that they give later in the paper, is that a labeled or radio-labeled phenylalanine would be given to the patient as a ingested diet, and then you'd see how they would respond to that before they were ever on the biotic, so day -1, and then when they were 2 weeks onto these agents, and to see a difference in terms of how they process or how they metabolize that labeled agent. They also looked in terms of seeing in terms of non-labeled phenylalanine, how those levels were kind of on 1 week and 2 weeks into the study, as well as that follow-up visit, which would have been 2 weeks after they would have been discontinued or stopped on the biotic agents as a kind of a follow-on period. They looked at the metabolites. We mentioned those earlier, the TCA and hippuric acid. So again, this is the kind of the other end of it. Are we seeing an improvement in the metabolism because are we seeing the increase in the products? And so they looked at these in terms of both in the blood and urine, both labeled. So again, as a product of the labeled phenylalanine as well as non-labeled on day 7 and day 15. The authors also performed as a kind of exploratory endpoint a Kent-TAB, which is a neurophysiological test on day 14. So this looks at things like spatial working memory, visual information processing, just to see was there any change over this 2-week period compared to a baseline or day -1. The study design was an open label study and was performed in multiple centers in 9 sites in the US. It was a 2-arm study, but the 2 arms were each represented the 2 different strains, and there was no placebo or kind of current treatment arm without any strains being given. That said, as we'll see in the further on the presentation, patients were allowed to, generally speaking, continue on their treatments as they would. This was an additional therapy. And the patients were non-randomized. The data was collected between February 2020 and October 2022. To be included, they had to be adults with PKU. They had to have a phenylalanine level of above 600 at screening, which would be kind of outside of ideal control level. So again, someone that you'd try to be targeting to see can you improve control. They have to have a stable diet before they were commenced on screening. And they could continue their Septarin or Kuvan if they were on it, but they had to have stable dosing. So they're going to be having dosing that was being changed or being adjusted before they went into the trial. They had to have a regular dose already set. The patients that were on pegfilase couldn't be included, so that wasn't the exclusion criteria, as well as those PKU patients who had conditions that would affect absorption of medication or nutrients, as well as thinking because this is a GI kind of targeted therapy with that in mind. Also, in the context of what we're doing here, or what the authors are doing here, is that antibiotics were prohibited kind of before the trial as well as during to the final safety assessment. They looked at any signs of adverse events, as well as vital signs and laboratory parameters during the study. And this is kind of 1 week in the lead up, as well as up to that 29-day follow-up. They obtained consent for participating, and they were compensated for their time in the study. The biotics basically were given in a dose ramp regimen, which meant that they started on, like I say, 1 dose per day, and that was gradually increased over the 15 days. And they're administered orally in the suspension of about 100 mils of water. The diet was set using a personalized food menus so that the patient would be maintaining their baseline diet, but they did record its kind of diet intake in the run-in period up to the safety follow-up visit. They were also put on a proton pump inhibitor as well from the start of the diet run-in to the last day of dosing, or day 15. The labeled phenylalanine, which in this case it was a deuterated phenylalanine, or radio-labeled, was basically conducted as an overnight fast till day -1, and then on day 14. And they took blood and urine samples cover the 24 hours to check levels. And additionally, they looked at unlabeled phenylalanine and its metabolites on day 1, 7, and 15. And before those, their patients received a breakfast of minimal phenylalanine, as they probably would normally, and then had blood and urine samples taken over the subsequent 4 hours. Ethical approval was obtained in the 9 sites where the study was performed, and the statistics that were performed were appropriate for the study. In terms of the results, so 28 patients were screened, of which 20 ultimately were enrolled, 11 in arm 1 and 9 in arm 2. Of note, about 5 patients discontinued in the 2 arms. 1 in arm 1, which was due to gastrointestinal anxiety symptoms, and 1 due to what they described as kind of failure to kind of comply with the protocol, the study protocol, as well as gastrointestinal effects, and 3 further with adverse events, 2 with GI symptoms and 1 with possible allergic symptoms. The kind of profile or demographics of the patients included are in the table here. They're roughly around 30 years of age on average, with a kind of roughly 50-50 male to female ratio. And their baseline levels were again around 900 to 1,000, which again are outside of what we target, which is up to about maybe 360 to 400. So getting into the primary outcome of the trial. So what they had seen was that compared to their baseline or day -0 levels, that there was a decrease in the phenylalanine seen in the blood on day 14 of the study. And that was both for arm 1, which is the light gray graph here, and arm 2, which is the darker column here, which is the second biotic that they use, the 1934. It might be a little bit easier to kind of see in the second of the two figures to explain. Each graph here represents arm 1 and arm 2 of the study. The orange dots, the orange bits of the figure, represent the phenylalanine levels seen on the baseline level or day -1. So you receive the phenylalanine tracer dose, the level will go up in the blood and then will be gradually metabolized and decrease. And that was seen in both arms. And when you compare that to day 14 on this biotic treatment, you'd see less of a peak of that phenylalanine and therefore less of an overall exposure to increased phenylalanine levels during that 24-hour period. And so that's what they're measuring, that kind of area under the curve, which was decreased by about 30 to 40% between the two arms. So that seemed to show a notable change. When they looked at unlabeled phenylalanine, again, a similar decrease was seen across the 2-week period. So between arm 1 and arm 2, they'd seen about, say, 15 to 40% decrease in the phenylalanine levels in the blood. At day 29, which is when they're kind of basically off treatment, it's kind of a follow-up review, you can see that the levels kind of go back to roughly their baseline levels. So they, in other words, after 2 weeks without the biotic, things kind of go back to how it was before. When they looked at the metabolites, what we're expecting to see is that if this is working well, is that we're seeing more of the metabolites seen because it's getting through processing more phenylalanine. And that's essentially what they've seen in the study. So these two figures represent the TCA and the hippuric acid in the blood and urine, respectively. Between the two arms, the blue being arm 1 and the orange being arm 2, they were all seen as increased compared to without the biotic day -0, which indicates that the body's metabolizing more of this phenylalanine compared to without the biotic in place. Perhaps unsurprisingly, there was no changes seen in the CANTAB or the kind of executive function scoring in either arm. It was over a short period of time, so I'm not sure that was unsurprising. In terms of the safety results, there was no serious adverse events reported, so that should be Symphony 1, not Symphony 2. The tyrosine levels remained in the safe range for all the participants, which is what we want to see. We don't want to see that they're deficient in tyrosine either. But that said, there was a kind of notable number of mild or moderate adverse events, which are predominantly gastrointestinal, so nausea, abdominal pain, diarrhea, etc. And 4 of the participants discontinued because of these in particular. The authors comment that in other studies that have looked at synthetic biotics or biotic therapy, gastrointestinal effects are the most commonly reported adverse events that are seen. So this doesn't seem to be unique to the study, but it seemed to be an issue that came up similar to other studies that have been done for these kind of agents. In terms of the authors' kind of comments on the findings, that they seem to show that both the biotic therapies seem to lower phenylalanine levels for patients with PKU, and that seems to be because they're metabolizing more of it within the gut. That said, they did see that their baseline kind of phenylalanine tolerance was seen to be better, and that they were metabolizing more of the phenylalanine even when they were not getting a dose in the diet. And this kind of, they hypothesized that perhaps that the gut is kind of access to the pool of phenylalanine that's in the body. So in other words, even though the phenylalanine is in the bloodstream, that perhaps the gut is able to kind of have access to that and break that down rather than being kind of like a solid space that once it gets digested and absorbed, the gut can't interact with it or be involved in that process. The authors acknowledge a couple limitations, which is that the number of withdrawals that they saw from the gastrointestinal adverse events, the open label design that the study had, as well as the small sample size that was recruited. And then potential challenges to this kind of therapy is as mentioned, the gastrointestinal intolerance. The fact that, if this is an Asia that if you're on antibiotics, that you're going to see reduced or incomplete activity seen because it's a E. coli agent. And at present, there's not any kind of examples of a synthetic biotic being kind of widely approved or widely used. So this is a kind of a new direction or a new step in terms of for potential therapies. In terms of from kind of our impression of the paper is that it did seem that the biotics led to a reduction or was associated with a reduction in phenylalanine levels at a notable amount. That the multi-center design was useful in the sense that rather than being performed at one site where the practices there could be an impact or a variable in terms of the outcomes, that having in multiple sites kind of dampens that possibility. And that the use of the labeled phenylalanine as the kind of primary outcome, I thought was useful because it's very defined a metric. The negatives was that just the high dropout rate is about 20-25%, which was notable, and due to side effects, which is going to be an issue long term if this is going to be something that you're going to look to try and get through phase 3 and subsequent clinical trials. That the fact there was no placebo or kind of comparison arm for the study to compare to is another issue, and the open label design was a factor in terms of expectancy observer bias. But that said, the use of the choice of primary outcome as a biochemical marker reduces the likelihood of that being a factor compared to, say, if it was a more of a subjective measure like pain or a pain improvement as say an outcome measure. In terms of the future for synthetic biotics, so the plan was to go forward with a phase 3 trial using the 1934 biotic because that seemed to have a better efficacy. And that was commenced, but unfortunately, however, that study was concluded because it didn't seem like it was the interval review seemed that showed it wasn't going to meet its primary endpoint. So unfortunately, that's on pause at the moment. But that said, there's certainly a promise here in terms of synthetic biotics as another avenue. And the authors comment that PKU, if it works PKU, there's a number of metabolic conditions that this could be beneficial for as well beyond it. In terms of for the future of PKU, beyond things like synthetic biotics, there's other things we're looking at as well. So gene therapy and gene editing is something that's an ongoing work. There's a good review article published in January of this year, which looked at all the different kind of types of gene therapy that are being reviewed. There's currently clinical trials for the adenovirus-associated gene addition, but we're waiting on the results of those at the moment. So I suppose in summary, this phase 2 trial seemed to show that synthetic biotics led to a reduction in phenylalanine levels. I suppose the issue here was that it was a small cohort without a standardized treatment or placebo arm. But I suppose the study was a useful example of that. It showed the challenges of performing a trial for rare diseases. So despite the fact that it was performed in the US across 9 sites, they ultimately only recruited 20 patients. So I suppose it's very difficult to get the numbers that you need to try and pull out strong results because of the fact that, again, the fact that it's a rare condition, the fact that getting patients to kind of sign up for trials where they're signing a very strict regimen in terms of diet is difficult. And so I suppose that's one of the challenges that I suppose a lot of us, the folks who are attending can comment on as well. And while phase 3 trials have been commenced, unfortunately, we haven't seen a successful completion of a phase 3 trial. But I suppose our takeaway from this was that it's a promising new avenue for potential therapies for metabolic conditions and specifically PKU. These are my references, but I just want to say special thanks to a number of people, Dr. James, Suzanne for organizing and helping me out with this, the metabolic team and the team at Temple Street, and just the Rare Disease Clinical Trial Network for this opportunity to present. Thank you very much, and I'm happy to take any questions.
Thanks, Kevin. That was a great overview of the paper. So if you have any questions, again, please put it in the Q&A box. I might just start off. So it's great, and it's interesting. So two questions. The first is putting your kind of pediatric hat on. Obviously, this was over 18s, and as you mentioned, sometimes you've missed the boat by the time they're adults. If they have PKU and the metabolism and the neurodevelopmental outcomes. And with children, is there any sign of using this or subsequent things in children? That's the first question. The second question I have is, obviously this is a very short study over 2 weeks, and they're using a daily treatment, which obviously has compliance issues. Would there be any thought of having a trial of something which is, a once a week or something that would be more compliant over a longer period of time? Because obviously this is something the person theoretically should be on lifelong, I imagine.
Yeah, both good questions. So, in terms of using something like this in pediatric patients, I suppose from speaking with our team in chemistry, we're interested by these kind of studies. I think where we are is kind of looking to see how they get on with adult trials, before, I suppose, seeing the potential of using them in pediatric patients. I suppose it's similar in terms of for any kind of pediatric new therapies or clinical trials, is that often you see how they get on it in adult medicine, and if things seem successful, someone then takes it on to see if they could do a trial for pediatric. So I don't think there's any. Look, I can only speak for chemistry. I don't think that I think we actually, if people have been approached, I think, in terms of about trials like these, but at the moment there's not any kind of plan for doing a, say, biotic trial for pediatric patients in Temple Street. To the other question, which is, I suppose, the issue of compliance and the fact this is it's actually a multi-day regimen. It was 3 or 4 times a day, and the issue of compliance for that. I suppose ideally what you'd want is something that you would take, as you say, once a week or once a month, and would kind of last. And I suppose, with something like an E. coli strain, would you look at something like you basically become colonized with it? But I suppose in previous papers, the authors kind of commented of a bit of a balancing act in the sense that this is a genetically engineered bacteria, and the risks in terms of basically something that you give that people will then be colonized with it and walking around with. Now, one of the things that they do to mitigate that is that the E. coli strain is autologous, which means that it can't survive in the wild, and so that prevents the potential spread of a genetically engineered bacteria. I do wonder in terms of if there's a bit of a financial, kind of a calculus to it in terms of that. If you could just give someone that they become colonized with, and then they just they maybe need a top-up dose once or twice a month, that wouldn't be as financially financially viable maybe. But I do wonder if that balancing fact is there as well, in terms of that, people being worried about genetically engineered bacteria getting into the environment, being a factor. But that's my thoughts.
Thanks. Thank you for that. There's one question. So Roberta Mendez, which I'll just throw out to you. So very good presentation. Thanks very much. You mentioned about additional dietary monitoring because the biotics can have the action modified by the diet. Is that correct? And accordingly with the diet, the metabolism of nutrients in the gut can be different. I'm curious to see the effect of the biotic in a long time treatment.
I suppose the first part of the question, which is the fact that the patients had their diets kind of monitored, or you might even say kind of regimented during the 2 weeks of the trial. It's not maybe too unsimilar to how PKU patients' day in day is, in the sense that they have to be very strict in terms of their intake of protein. They kind of measure their exchanges, or how many grams of natural protein they're getting in their day. I think part of it, though, was the fact of to try and keep it a strict control for the fact of being in a trial, and so that they could basically comment that that the patients were being kept to their regiment, and that they weren't kind of having wildly uncontrolled PKU during the trial. But that said, it's not unsimilar to what the usual day in day is for PKU patients. To the other bit, which was looking at more long term in terms of the effect on the diet, it's an interesting question. And I don't know in terms of what say this kind of biotic therapy would have in terms of for a PK patient after say 6 months. I suppose we'd have to do the trial to see, or a trial would have to happen to see. But I actually it'd be hard to say until you looked at it. But yeah.
Great, thank you. Is there any other questions that anyone has? Yep, Rachel.
Thanks, Kevin, for a great presentation. I was just curious, for people living with PKU who aren't on these therapies, are there differences in their gut microbiome compared to people who don't have PKU, or is there a different metabolic signature from their gut that might explain why this is such an effective therapy potentially for this group?
That's a great question, and the honest answer, I don't know. I'm not sure if I've seen anyone look at the gut microbiome for PKU patients specifically. It's an interesting question. I know that the choice of the strain for this study was chosen because a non-synthetic form, so one that didn't have any genetic engineering, has been used previously for patients with like inflammatory bowel disease or IBS. And so that was the thinking behind it being kind of a safe agent. But I think the idea that they're proposing is that, with the genetic engineering, the addition of the phenylalanine enzyme to metabolize, is that's where the change or the improvement is being seen. But it's worth asking in terms of like, does the microbiome have an impact in of itself? I don't know. It's worth looking at. I'm not sure if there's any publications on it, but I have to look.
Thanks.
Just on that, so Roberta just had commented, so she's worked in a research project evaluating the gut microbiome in PKU and control. So in the middle of doing that, so she's going to add a link to that paper. So we'll add that onto. It's like a planted question, so she'll add that onto the link, and we can forward that on to people if they want it.
That's fabulous. I suppose that's one of the benefits of these kind of presentations. I get to find out these kind of things. That's great.
Yeah, and I mean, the last thing, I mean, if there's other questions, happy to, but it'd be interesting to see what the phenotype of using this would be long term, because obviously that wasn't, it was too short. And like you said, when they did the questionnaire, I mean, I don't know if you're ever going to find a difference 2 weeks afterwards in a questionnaire, but it'd be interesting to see what, a year later and phenotypically, is there a change? Because obviously that's the main endpoint, isn't it? So it'd be interesting to see that.
Yeah, true. Yeah.
Okay, maybe for the sake of time, so thank you very much again, Kevin, for that. We might move on to our second presentation. So our second presentation is Dr. Bridget Moran, who's a ophthalmology SHO in the Mater, and she's going to be giving us a talk of an overview of Stickler syndrome. So Bridget, you're very welcome.
First of all, I just wanted to say thank you so much for this opportunity to present to you today. And I also wanted to thank Professor Gaughan for helping me select this paper. So today we're going to be talking about Stickler syndrome, a review of the condition and of a proposed care pathway here as based on a similar pathway seen in the UK. The paper that was reviewed as part of what was included in this review was a paper on dominant Stickler syndrome by So et al. And also included here was the NHS model of care. Yes. So just some background information about Stickler syndrome. The condition was first identified in 1965 by Gunnar Stickler, first recognized as a condition affecting the eyes and the major joints. But as time went on, it was recognized that it had multi-system manifestations affecting the eyes, the ears, the facial development, as well as the musculoskeletal system. And it has also been discovered through epidemiologic studies that it is the leading cause of inherited retinal detachment worldwide. And of interest to us as well is the incidence of this condition and how there may be as many as 500 cases occurring in Ireland alone. So how does Stickler syndrome present? Well, there are many manifestations. As I was saying, there are auditory, musculoskeletal, oral, orofacial, and ocular presentations. So these patients tend to present with high myopia. By definition, this would be myopia of -6 diopters or higher. And this myopia places them at significant risk of retinal detachment, especially of giant retinal tears, which are defined as retinal tears which are exceeding 3 clock hours. In addition to the myopia and the retinal detachment, some manifestations that Stickler syndrome can have in the eye include cataract, strabismus, astigmatism, and glaucoma. And here are some of the images depicting the manifestations. So these two first images depict giant retinal tears, and we also have some images here of the classic phenotypic presentation in children, the mid-face hypoplasia, flattened nasal bridge, and myopia. And pictured here are some of the classic vitreous manifestations, anomalies that are seen in this condition. Going further into the ocular manifestations of Stickler syndrome, it is a condition that can present in the eye as early as infancy, and it may only be detected once the eyes and ears of neonates are examined. And it is because of this high myopia and these vitreous degenerations that this population is at such a significantly increased risk of retinal detachment compared to the baseline population. In addition to the increased risk of these retinal detachment and multiple more than would be seen in the baseline population. And in terms of the genotypic features of this condition, it is by and large an autosomal dominant inherited condition with 100% penetrance, meaning that features either manifest or don't. And it is also worth acknowledging that there is no race or gender predilection, and that there is no impact on life expectancy or mental capacity of those affected by the condition. Type 1 Stickler syndrome accounts for the vast majority of cases, and it occurs due to pathogenic variants of type 2 collagen. This type of collagen is the most abundant type found in the vitreous. And it is worth noting that type 1 can present with multi-system findings as well as findings confined solely to the eye. In addition to COL2A1, which causes type 1 Stickler syndrome, variants of COL9A1, 9A2, 11A1, and 11A2, which are responsible for the formation of type 9 and type 11 collagen, respectively, can cause other subtypes of Stickler syndrome. And more recently, BMP4, which is bone morphogenetic protein, and a loss of function mutation in this gene has been found to cause a Stickler syndrome phenotype, which also affects the kidneys and the urinary tract. Shown here are the major gene loci for these three types of Stickler syndrome. On chromosome 1, type 2 Stickler syndrome, which is affecting the type 11 collagen, is found. As well, on chromosome 6, there is a gene loci which affects collagen type 11, and then type 1 Stickler syndrome, which is by and large the most common, is affected by mutations on chromosome 12. So taken directly from the paper by So et al., we have a table compiling the major types of autosomal dominant Stickler syndrome. Type 1, which can be multisystem or ocular only, is the major contributor to blindness associated with Stickler syndrome. It is the subtype with the greatest risk of blindness and of retinal detachment. Type 2 and 3, which affect type 11 collagen, are also contributors, but it is type 1 that is the most common and 2 of the highest risk when it comes to retinal detachment. And type 7, which is due to the mutation in bone morphogenetic protein, has yet to be quantified in terms of how many are affected by this subtype relative to the others. So in terms of the diagnostic criteria for Stickler syndrome can be diagnosed using genetic testing to identify the variants we've discussed already. But before this testing is done, it can also be diagnosed on the basis of findings when it comes to examination of the face, the ears, the eyes, and the musculoskeletal system. So in terms of orofacial abnormalities, there are major and minor signs which could suggest a diagnosis of Stickler syndrome, major one being cleft palate. Some of the patients who are diagnosed with Stickler syndrome have Pierre-Robin sequence. A sequence is a condition which gives rise to other conditions. In the case of Pierre-Robin, they have hypoplasia of oral facial hypoplasia, which causes the tongue to protrude backwards, which in turn affects the development of the palate, in some cases causing cleft palate. Some of the features which are less strong of an indication but still point towards this condition include malar hypoplasia, the broad or flat nasal bridge, and micro or retrognathia. The ocular abnormalities which may be present include vitreous changes, such as retinal detachment or retinal tear. In terms of skeletal abnormalities, features such as femoral head failures, such as Legg-Perthes disease, and radiologic demonstrated abnormalities of the joints, especially of the spine, can also be suggestive of Stickler syndrome. So the molecular testing, as we discussed, is worth 1 point in this diagnostic criteria that was proposed by Rose et al. Is only worth 1 point and does not count for families with autosomal recessive Stickler syndrome. So finally, the auditory abnormalities. So while Stickler syndrome can cause sensorineural or conductive hearing loss, it is only the sensorineural that is given points in terms of the diagnosis. It is worth noting that these 5 points that could be what is needed to diagnose Stickler syndrome could be made up without the ocular signs. However, the vast majority of these cases would present with ocular signs. And it is worth thinking about what cohorts might have Stickler syndrome in terms of what other signs they might present with before the myopia is recognized. So the Pierre-Robin, as we had mentioned, may be the first aspect that is recognized on the child, or it may be the myopia, which is out of proportion for the age of the child. As a general rule of thumb, it is suggested that if the diopters of myopia is greater than the age of the child, that this would be disproportionate. And it's also worth considering a family history of joint hypermobility, midline clefting, or deafness, even in the absence of a formal diagnosis of Stickler syndrome. So what do we know about the prophylaxis of retinal detachment in Stickler syndrome? Well, we know in recent years that this has been investigated and the methods of prevention have been evaluated and compared. One of the methods with the strongest evidence bases behind it is cryotherapy, as put forward by the Cambridge Protocol. This involves 360 degrees around the perimeter of the retina of cryotherapy, and this has been shown to prevent giant retinal tears, to the extent that when patients who had this cryotherapy were compared to ones who hadn't, there was a 7 times lower risk of giant retinal tears in the cohort that had the cryotherapy, especially relative to those who hadn't. Even when the eyes of participants who were treated were compared to those, when the fellow eyes of participants were treated with a treated eye versus an untreated eye, there was still a greater risk in the untreated eye of developing retinal detachment. To some extent, laser is also being put forward as a prophylactic measure. However, the evidence base is smaller. Here are some photos demonstrating what the cryotherapy looks like. It is circumferential around the periphery of the retina, where the tears are most likely to occur. These images all depict the cryotherapy. Here, it's all visible around the periphery. And this last photo indicates an area of retina which was treated with prophylactic laser. However, the tear did occur in this case. So leading into the NHS model of care for Stickler syndrome, given that it is a leading cause of inherited retinal detachment, especially of retinal tears, concerns were raised about the degree of blindness that would arise from these tears, especially due to type 1 Stickler syndrome. And as a result of this, a multidisciplinary service was put together, including ophthalmology, rheumatology, audiology, and maxillofacial surgery, to identify this high-risk patient cohort. In addition to this, a registry was kept of patient data in order to aid the further assessment of suspected cases, to assess the outcomes of prophylaxis, and to determine the extent of multi-system complications associated with this condition. In patients who had phenotypic criteria that led to suspicion of Stickler syndrome, they were referred to this multidisciplinary clinic and examined for the characteristic features which we have discussed: the ocular, auditory, musculoskeletal, and radiologic features of this condition. And if this exam was found to support the diagnosis, genetic testing was done on blood samples to look for the collagen mutations in collagen types 2, 9, and 11. There was also testing of relatives carried out, and if results were discussed with the patients, counselling was provided, and any cases that were unaffected were discharged from the service. So further into detail about the specialised service offered by Cambridge, it is a service which is multi-specialty. It revolves around 3 clinics a week, which currently serve patients affected by Stickler syndrome, both in the UK and in Ireland. However, we're unsure exactly how many patients from Ireland are travelling across for these services. But we do believe that a similar model could be implemented here in Ireland. So just delving further into all of the elements of the multidisciplinary team that would be needed for this condition, we have ophthalmology, which are essential in regularly examining these children given the risk of retinal detachment, for providing the cryotherapy, the prophylactic cryotherapy, on which so much evidence is based on. We also need pediatrics who are essential in identifying the early signs of this condition, given how early it can present and the downstream developmental effects of retinal detachment, such as deprivational amblyopia if the vision were to be impaired at a critical stage. In addition to this, rheumatology would be vital in terms of protecting these children's joints and facilitating rehab. In a similar role, orthopedics will be essential. ENT and maxillofacial would be important in taking care of the oculofacial complications, and in those, especially with the BMP4 variant, nephrology have a vital role. So in conclusion, Stickler syndrome is the leading cause of inherited retinal detachment worldwide and in Ireland. There may be as many as 500 cases suspected on the island of Ireland. However, far fewer than this have been identified here. It has been demonstrated by the Cambridge service that a bespoke pathway of multidisciplinary teams could aid in early case identification and diagnosis, in addition to providing effective prophylaxis against retinal detachment and any resulting blindness. And a multidisciplinary approach would provide the best comprehensive management of any comorbid conditions. Despite the available expertise and resources, such a pathway has yet to be implemented in the care of Irish patients. However, we feel that it would be feasible given the resources we have here. And these are my references. Thank you for this opportunity to present today.
Thanks, Bridget. Thanks for a good overview of Stickler syndrome, both the paper and also the NHS guidelines. So again, please feel free to put questions into the Q&A box, and we'll share that with the audience. So I might just start off, I guess just an overview. You mentioned there's no dedicated service in Ireland at present. Are the Stickler patients seen are they kind of cohorted into one or two main centers at the moment, or is it just kind of a ad hoc throughout the country where there's just pockets of the patients? Are you aware of what's kind of happening in Ireland at the moment with these patients?
From an ophthalmology standpoint, there is a specialized inherited retinal degeneration clinic which runs in the Mater Hospital, which provides an overview and provides additional support to patients with Stickler syndrome. However, this hasn't been coordinated into any large sort of multi-specialty clinic. However, all of the specialties that I've mentioned, the ENT, maxillofacial, rheumatology, are all present here in the Mater. So that would help to keep things all under one roof if it were to be put into place.
Could I mention the children? Could I maybe the echo?
Yeah.
I might just come in, Adam, just a little bit about the children that Bridget is less familiar with on the pediatric side, because it is, you are right, there are services for Stickler syndrome between Crumlin and Temple Street and the Mater Hospital, and some patients will be seen over in the Eye and Ear as well, but there's no coordination of that. So what we're doing under the Inherited Retinal Degeneration Programme, start to bring those together. We haven't shut down the transfer of patients abroad to Cambridge, but we know there aren't anything like that number of 500 patients going for regular cases over there, and there aren't that number of patients on our books here. So before we go and extend that screening testing out, and it's interesting, listening to Kevin's talk about screening for PKU, which is so well established in that this whole concept of screening for genetic disease is now going to sort of come in, but we have to have the service available. This is my ongoing discussion with Richard Hagan in the National Genomics Office, is you turn on that tap, you've got to be prepared for what's going to come out. So we're trying to get ahead of it with a very clear index condition. And I know it's not really part of the clinical trial. So Bridget presented the clinical trial data or the clinical outcome data from just a very simple intervention. It's not gene therapy, it's not biologics, it's just freezing the retina, reducing the retinal detachment risks by 7-fold. So again, it's pushing that concept of integrated care. So we've got expertise across, we've got expertise in Vincent's and Crumlin in the Mater and in Temple Street, how we pull all that together to recreate what they have in Cambridge. But it's hard to get a full handle on the numbers. And this is where I'm going to put a little bit of my advocacy political hat on the amount of money being spent through the Treatment Abroad scheme. going to Cambridge for these children, because that's where all the applications are filled in from. So we've an ad hoc, but we're starting to provide the treatment. So we're now doing the cryopexy treatments in Ireland. I've commenced it, but not at anything like the level that we need to get it to. I hope that answers your question.
Yeah, no, that's great. Yeah, and I mean, it is, with, especially in rarer conditions, I mean, that MDD is such a vital role. And like you said, you have to have it there. There's no point diagnosing if you can't have the MDT and treat. And I mean, I'm pediatric cardiologist, and the only thing with, obviously the Ehlers-Danlos and Marfan's or Lois Dietz that we come across that then we're referring, but at the moment, similarly, it, for a lot of it in the pediatric world, you're seeing certain subspecialties, but it's getting kind of that cohesiveness of care and all in the same room. And, it's important from the patient impact standpoint. So yeah, absolutely. I think it's great that seems to be coming together somewhat.
Very early days.
Yep.
So many rare conditions, I think. But I think it's a good index condition for us to really work on. I think it's well established going abroad, and and we can maybe put a cost on it, and we have the demographic data that we should be looking after here. So I think we can work up the case around it. Yeah.
I mean, the hard thing, like you said, is that the money for the treatment abroad scheme is coming from a different purse than the money for the clinical thing here, and trying to get the powers that be to understand that it's cost neutral, probably, if not cost beneficial, but it's coming from a different source sometimes can be harder to do.
Yeah, you can't necessarily access it.
No. Any other questions from the audience?
I could add a comment while you're waiting, Adam. So I think we can clearly see in this case that cryotherapy, like freezing the retina, is an intervention that's sort of obvious to us. It's something that you did that was potentially preserving sight by preventing a tear. But actually, sometimes bringing in a new service delivery is an intervention that we can measure as a trial. So the very classic example that's been used in Ireland repeatedly is if you have a proper diabetic foot service where a patient gets a podiatrist and a vascular surgeon and infectious diseases involved up front, you can prevent amputations. And it's actually not a new medication or a new surgery or anything that we classically think of, but it's a way of delivering the service. And that can be an intervention that we can trial. We can do a before and after study on that type of thing.
Okay, because actually I was quite conscious, Rachel, about, I know it's a clinical trial network, but you can see what we were looking to do by bringing this to today. And thank you to Bridget for pulling all that together. So actually, I hadn't even sort of considered about that intervention as a trial, and we'll do that. And it's something we can maybe work with Carolyn on. It's sitting right under our nose. And actually, with Adam on the call, the other thing I'd spoken to Mark Redmond, Adam, about his adult Marfan's clinic, which he runs quarterly, all day Friday, 4 times a year. So we're going to just rearrange our eye clinic just so the patients come once. We improve the attendance rates. So just try and coordinate it like that.
Yeah, I think, and I mean, that's part of what I guess what this whole thing is about, is trying to bring people together and, try and coordinate things. And because we're all in our sometimes in our silos or different areas, and obviously it's better if we can pool some knowledge and resources if possible.
So, well, that's the key, Adam, when I go on too political on it, is that we can pool resources, that if we go out to set up a new pathway, that there won't be everybody going, I need a full new service, I need a new nurse, I need a new administrator. I say, look, you're seeing these patients anyway. Maybe can we just do in a coordinated fashion, that we're maybe looking for one central coordinator rather than building 5 new teams. But if we can get consensus on that from this group in this environment, it'd be great. Without taking away from that frontline innovative stuff that we heard from Kevin earlier on. I mean, that's what we've got to be pushing that. But how we can advocate for both trial models would be great.
Okay. There's no further questions, Simone does say, excellent point, Rachel, just to Rachel's point that, but I think if there's no other questions coming from the audience, I'd just like to again thank Kevin and Bridget for their presentations. Both very, different but very valid presentations, kind of, I guess, like Dave said, two parts of the spectrum. And I thank all the participants for coming. We are going to be just going forward, and Suzanne, I might let you ring in at the end, but we're going to try and keep this going up, and we'll post it on social media and through various channels about when the next journal club is happening, with 2 different presentations. We would like everyone to please feel free to come back for those, get involved in the Clinical Trials Network. Thank you very much for coming. Rachel, is there anything else you want to add?
No, we just noticed how varied the attendance from people is today. So how many specialties and backgrounds are represented in patients. So it's great. You're very welcome. Please spread the word to your networks, as Dr. James is saying here, and we value your contributions and your thoughts. Thank you very much.
Suzanne, I'll let you close. If there's anything else?
Yeah, no, I don't think there is. And thank you for that. And thank you, Adam, and to the speakers and everyone on the panel and the participants. And as you have said, watch this space, because now that we have the Journal Club up and running again, we hope to have it as a regular feature. So thank you.
Okay. Well, thanks very much again, Kevin, Rachel, and have a good day, everyone. Thank you.
Thank you. Take care. Thanks, Kevin. Take care.